Hello everyone, my name is Angela Echeverry and today I'm going to talk about an article that calls Emergency and Characterization of Nosocomial Multidrug Resistant and Extensively Drug Resistant Acinetobacter baumannii isolates in Tehran, Iran. First we need to talk a little bit about the Acinetobacter. This microorganism is a Coccobacillus gram-negative bacteria. It belongs to the nosocomial pathogens group, which means there is a bacteria that you can find and get infected in a hospital or other healthcare facility. Of course, it only happens when there are unwise managements of the environment. Also, this bacteria is associated with some disease like pneumonia, bloodstream infections, meningitis, necrotizing fasciitis, and urinary tract infections. Because the Acinetobacter is one of the antibiotic resistant that threatens hospitalized patients, we need to talk about the antimicrobial resistance, which is very simple. It's the ability of microorganisms, bacteria, viruses, and some parasites to stop an antimicrobial, such as antibiotics, antivirals, antimalarials, from working against it. As a result, standard treatments becomes ineffective, and the infection persists and may spread to others. Now, knowing this, we can talk about the objective of this article, which is try to characterize the antimicrobial resistant profiles and the genotypes of clinical and the environmental isolates of A. baumannii as a result of dissemination of resistant strains. Now, the material and methods. This study used 4 methods. The first one is the bacterial isolation. This technique used to transfer a microorganism from one environment to another, was used in this study to induce the growth of the microorganism for its identification. This technique consists to collect environmental samples very carefully using a wet swab from clothes and hands of staff, medical equipment, and patient's environment. Then all the isolates are going to get cultured on a McConkey and blood agar instantly after sampling and then incubated at 37 degrees for 24 hours. For the last, with the help of some biochemical methods, we identify the A. baumannii. The second method is the disk diffusion method. This technique used for determining the antimicrobial susceptibility of some microorganisms. We use to define the susceptibility of the Acinetobacter baumannii. In this technique, wafers containing antibiotics are placed on an agar plate where bacteria have been placed, and the plate is left to incubate. If an antibiotic stops the bacteria from growing or kills the bacteria, there will be an area around the wafer where the bacteria have not grown enough to be visible. And it shows that the bacteria are not resistant to antibiotic. Contrary, if the bacteria grows, it shows the opposite thing. And for the last, the third and the fourth method, PCR, multiplex, and repetitive. Although PCR, multiplex, and repetitive are different techniques, they have the same fundament, which is that they use the polymerase chain reaction to amplify different target DNA fragments, and we use this technique to determine genetically which antibiotics are resistant to A. baumannii. So, the PCR multiplex consists of several sets of primers within a single PCR mix to produce amplicons of size that are specific for several different DNA sequences. The PCR rep is a PCR amplification of repetitive extragenic palindromic elements, a type of PCR that uses primers designed to advertise with repeated sequences interpairs in the genome. Now, results. The first table that we have, the important thing here is that it shows the genetic elements that were identified in the PCR multiplex and repetitive methods. This graphic is called a dendrogram, and it shows the percent of similarity from one bacteria to another one. Very important in this study because we have the Acinetobacter isolation, but we need to see if they are all the same or not. So, if the rep types are close to 100%, like the ones with the rectangle blue, it means they are similar. But if they are far from the 100%, like the one with the rectangle red, they are different. Also, we can see little differences between the rep types that were close to the 100%. For example, on one of the green circles, we can see a tail that on the other one, we can't. Or on one of the orange circles, we can see a line that on the other one, we can't. And we can explain this last thing because if we see the site of isolation and the word of isolation, we can see that are different between them. For example, the ones are inside the rectangle blue, which belongs to the group A, or the ones are inside to the rectangle red, which belongs to the single type group. They have different losses. There is another table which the important thing here is to see that there are some antibiotics close to 100%, like the one with the color red, which means they are very resistant. But there are also another antibiotics far from 100%, like the one with the color green, which shows that they are not very resistant. And the last table also shows the bacterial resistance. So the important thing here is to get that there is a high level of a bacterial resistance. So the question is, how are we going to treat these patients with this huge level of resistance? Discussion, there were some others who talk about the results that we were talking before. So for example, Senati K and some others said that A. baumannii is principally remarkable for its propensity to turn out to be MDR and XDR worldwide, specifically in the developing countries. And there is a happy face because it's something, what Senati said was in the article. So that's the happy face. Also, Lei G and others also says, in the present investigation, we firstly assessed the resistance profile which demonstrate high ratios of resistance except for colistin and taxicline. These two effective antibiotics play a major role in determining XDR isolates and have synergistic effects with other antibiotics families against drug-resistant A. baumannii, even though colistin is nephrotoxic and neurotoxic. And there is a negative sign because although there are some things that were on the article, there are others that they were not on the article. And the last one must be H and other ones, another authors, says that a majority, 97% of the collected isolates were considered as MDR strains, which was closely similar to the incidence of MDR strains in the other reports in Iran, 94%. There is also a happy face for it because what he says was in the article. And for the last, the conclusions. The first conclusion that we have is that if we promote therapeutic policies and prevention programs in the control of nosocomial infections, we can reduce the dissemination of some of these pathogens. And the second conclusion that we have is that this study highlights the circulation of drug-resistant A. baumannii strains in different wards of hospitals, principally in intensive care unit, as nosocomial pathogens due to unwise management. Okay, this is everything for today, so thanks for watching my video and good night.